how to use phylogenetic tree to understand character evolution. Has anyone of you thought about morphological characters, behavioral traits, and how have they evolved in whatever model systems you have working on? Or have you guys used any of these methods or thought about it, used it before? Okay. So whenever we think of traits, we could think about phenotypic traits, which is how you measure morphological traits, or it could be continuous trait or a discrete trait. It could be behavioral traits, if this particular species shows this behavior or not, or actually you measure the behavioral trait, or it could be functional or ecological traits. So any such trait, how that particular trait has evolved, is it the evolutionary legacy or historical contingency or a legacy of its history? Like because all the species shared ancestor had that trait, so it just passed on because the ancestor had that trait, or is it an ecological adaptation and that's why we see a particular trait in a particular species? So if one wants to understand that, this is where phylogenies can be very useful. You can easily see this part you can understand, but once we give a phylogenetic context when we understand the trait evolution, you can then think about, is it because of the evolutionary history or is it an ecological adaptation in that particular lineage? So all of you are familiar with this. What is the phylogeny? It's the hypothesis how species are related to each other. I'm using the word species, but whichever scale at which you want to think about traits, you can think about phylogeny of that scale. It could be at the genus level, family level, order level, whatever, wherever that trait is consistent within that particular group of taxa. And then, what is the aim here? To look at the significant association between those trait values which are at the tips and their phylogenetic structure, and then try and think how this particular trait has evolved. Yeah, that's the whole idea here. And under the trait evolution, you can have multiple hypotheses or multiple ways of thinking about it. So one is ancestral reconstruction. What was the trait? Right now in two species, trait A is present because their ancestor had it or not. So how do you do that? You do it with the ancestral trait reconstruction. But there are multiple questions you can ask. How have the trait evolution patterns? What's their influence on species diversification? Or which traits interact with one another and influence particular trait? So all these questions you can use and ask using phylogenetic framework. It's called comparative phylogenetic framework. You can use multiple traits. Today, what we will focus is only ancestral trait reconstruction. The methods for all of them are very different, so we won't get into that. We will just focus on how to reconstruct ancestral traits on a phylogenetic tree. So you have a phylogenetic tree. We have written that there is an uncertainty within the tree topology, but the methods we will discuss today do not account for that. We have a phylogenetic tree which depicts the species evolutionary relationship, but there are methods in which you can have multiple tree topologies on which you can reconstruct traits. So you have phenotypic trait data at whatever scale you want to think about trait reconstruction. So you have a phylogeny where all your species, genera, families, whatever unit you are looking at, and you have a trait data. And your aim here is to think about how this trait data has evolved in this particular phylogeny. So if I just divide the trait data in two colors, there is a blue here and a red. Easily, when you see a trait data, just by plotting on the tree, what would be your guess?
They should be phylogenetic.
Signal, but simply all blue traits may have a common ancestor, as well as all red, because see they have a very strong phylogenetic structure, right? This is a monophyletic group which has one particular trait. This is one group which has one particular trait. So by just looking at it also you can think about maybe this particular trait has evolved in their common ancestor here, the red trait evolved here, right? But this is the aim here, like you have a trait data which is at the tips, you have a phylogenetic tree and you are aim here is to understand how this particular trait in a given species has evolved, given this is the phylogenetic tree which depicts their evolutionary relationships. So how does one go about it? I'll just give you a couple of examples how people have used it, and then we'll talk about two such methods to reconstruct the tree. So this is the phylogenetic tree for a bumblebee. It's one particular group of bumblebees. Here, the authors were interested in trying to understand this one particular behavior which bumblebee shows is communal brood rearing. So they had the data for all the tips here. The species participate in a communal brood rearing or not. And when they did the ancestral trait reconstruction, they found the ancestor for all these bumblebees had a trait which was inherited by all the species. So ancestral trait was communal brood rearing. There are few species where this trait was lost, but this was the ancestral trait. Then they looked at yet another trait where species become parasitic. They had two hypotheses. It has evolved and retained in some species, or it has evolved multiple times. What did they find? It has evolved 3 independent times. If it was because of the shared ancestry, you could have had a clade where all the 3 species which are parasitic have formed one particular group, right? But when they mapped it on the tree, they found it has evolved 3 independent types. Another example of a behavioral trait, these are the phylogeny of orioles. Orioles do show migration. Is there a way, Maitri or Kunal, do you know how to change the aspect ratio here? For the slide. So orioles show partial migration, some of the species show short distance migration, some species show long distance migration. So authors started with, is there any directionality to this behavior? A species becomes partial migrant, then short distance, and then long distance. When they actually mapped the trait on the oriole phylogeny, what they found, there is absolutely no such thing, there is no directionality. Migration as a behavior has evolved 5 times independent of each other. You can see short and a long distance, these are species pairs. But partial migration, which is depicted in blue, has evolved twice independently, and that has no directionality. Like partial doesn't then later on becomes short or a long distance species, right? Without this information, we can't talk about how migration has evolved or if there is any directionality. So phylogenetic tree gives you that important information, and when you map this particular trait, you know now. You can actually test this hypothesis. Yet another example of lizards.
I did not understand the directionality part of it.
So, yeah. So if you have this particular clade, these 2 species, so right now you can think about this as a sister to this group, this is sister to this group, right? If these 2 had a partial migration, these 2 short, and these 3 show long distance migration. What would be the ancestral trait reconstruction? Maybe partial migration evolved, and then you had short and long distance. So you can think about a path species would have taken and then test for that, if that particular thing is true.
That would be only if the trait has such
Pattern.
Pattern.
So that's what you do. You don't know how it is structured on the tree, so you first plot it and then you reconstruct the ancestral traits.
So for this, any of these traits we are talking about, so this data we put into the software or we manually later based on our things?
So we'll come to that, but you have to collate the data, and then any of the programs which we use, you can maintain it in Excel sheet, Word doc, or Word text file, whatever format you are comfortable with. Yet another example of plants. Here they were interested in understanding how the syncarpy has evolved. Do any of you work on plants? What is syncarpy? Fusion of the petals. And people have been interested in this particular trait to think about how this syncarpy has evolved. Because was the fusion as an ancestral trait, and then you had multiple individual carpels. So this is the family of Ananaceae. tree. And here what they show that syncarpy has evolved in this particular crane. So it's not at all ancestral. So ancestral trait is multiple carpels, independent carpels. So that was the ancestral trait, and a fusion evolved later.
So we can collaborate the data of molecular with morphological data for understanding which character is primitive or which is advanced.
Yes, not advanced or primitive, ancestral and derived.
Ancestral.
Yeah. So this is the tree topology which is based on DNA data. All of you are familiar with how to build a tree topology now, right? How to reconstruct tree, right? Using molecular data. You have some morphological data, some behavioral data, some distribution data. Now your aim is to understand how that particular trait has evolved over phylogeny. So you don't use that trait data to reconstruct the tree. The tree is reconstructed based on DNA data. Is that clear? Another trait authors were interested in, lianas. How did the lianas evolve? Was it one single origin for a liana, or was it evolved multiple times? And again the directionality. Only tree to liana or liana to tree also has happened. So this is where evolutionary relationships can be very useful. So they show that there has been reversals also. So you can get evolution of a lianas here, but then there is a reversal to woody plants, right? And that happens multiple times in the phylogeny. Here is an example of geckos, and they were interested in trying to understand. This is now a phenotypic trait, morphological trait. How the adhesive toe pads have evolved in this particular clade of geckos. This is a genus level phylogeny. And then again you have a data for the tips, and your aim is to reconstruct the tree showing how is the adhesive toe pads as an ancestral character, and the other types of toe pads are derived. So they have mapped those. So when we use the word mapped, what does it mean? How do you reconstruct these traits, right? Right now you have a trait data. You are interested, what is the node here? Species where they diverge from their common ancestor, what was the trait there? That's the aim, right? So what sort of hypothesis one can ask when you have a trait data and you have a tree? You can ask multiple such hypotheses. Is the trait conserved, or is it because of conversion? Is it some under an ecological pressure that particular trait has evolved? Polarity or directionality of character change. What was the ancestral form? Partial migration to short and long distance, or vice versa. If more characters have stronger association and they influence each other and their evolution, can we study that? What evolutionary processes would lead to that particular pattern? If I have a hypothesis that if a species or a group of species under strong sexual selection, trait evolution should be like this, I want to test that hypothesis. This is one way to look at the data. So these are the different kinds of hypotheses one can use when you have a morphological, behavioral, or any other trait data, and try and understand how those traits have evolved in those group of species or genera or family. What methods right now do we know? There are, as usual, parsimony methods, likelihood methods, and Bayesian methods. I think all of you already know now how to use parsimony, likelihood, and Bayesian to reconstruct tree topology, right? So the approach or the principle of these methods, all of you are aware of, right? Similar principle now, but what is aim here is to reconstruct the particular trait and not the tree topology, but we use the same principle. We'll today talk about parsimony and likelihood, not Bayesian, but I can give you resources for that. We'll start with parsimony. Okay, before that, as we discussed, there are two types of trait data. What is discrete data? Presence, absence, binary. 0, 1. Present or absent, yes or no. So you can discretize, or a categorical data, right? 1, 2, or multi-state data. These are all discrete data sets, or you can have a continuous data. measurements. If I'm measuring frog, snout to vent length, body length, all these are continuous data types. So you can use both to understand the trait evolution. You can discretize continuous traits if one has to, but these are different trait types and you can use both. Simplistic. Have any one of you collected trait data for your whatever set of organisms you work with?
I have collected continuous data from my plant for the morphological analysis. I have collected the bulb length, the bulb breadth, diameter, and escape length for my genus.
Okay, so you know how to collect those sort of data. You can actually make the measurements or you can look at the literature and gather the data. The important point is think about what scale you want to use the data. Where is the trait variable? Where is the trait conserved? Define your scale and then gather the data. So discrete data is simpler to analyze. Continuous trait data, you have to think about appropriate distribution. Can any one of you explain what is normal distribution?
Normal distribution has a mean with the particular standard deviations on both sides.
Yes. Then what is exponential? And then what is Poisson? How would you think that if I have to transfer my data, you mentioned that you have a continuous data which is snout to vent length, right? And now I want to compare that data across multiple species. Do I need to transform that data? Does it follow any of these distributions or not? Does it require any transformation, right? So if you have some traits which are evolving discretely, is a frequency, you can think about it. Will you use Poisson? Do you understand what is the variance associated with your trait data and how that is structured? Based on that, you will think about what is the most appropriate distribution for your trait data. And then you need to transform your data. One of the most common ways is log transformation or a continuous data. To make sure it's normalized and it's usable. So it's comparable across species. So think about it whenever and whatever type of data set you have. For a discrete data, it's simple. Is it binary? Then it's fantastic. 0, 1. Sometimes it's multi-state. Sometimes you need to categorize it. Discrete, It's a continuous data. Think about what's the most appropriate distribution which fits your data, which explains your data. How is the variance structure in your data set and transform accordingly. So we'll discuss today one method in detail. Second method, I'll explain how it is used, and Bayesian, I'll give you the resources. Yeah? So all of you know parsimony approach to build a tree, right? Phylogenetic tree. What's the principle here?
Minimum number of changes.
To explain the relationship. Yeah? Same thing. The algorithm today we'll talk about is called Fitch-Wagner parsimony approach, which uses the same principle. Minimum number of steps should be able to tell you the trait distribution what you see in the phylogenetic tree. It's the simplest approach to think about fewest evolutionary steps to explain the trait distribution on your phylogeny, and branchlets are not incorporated in this approach, and we'll come to that in a minute. So imagine this is your tree topology. You have A, B, C, D, E. These are your species, and open and black circles are your trait data. And my interest here is to understand how the open and black circle have evolved in these two species. Imagine this is a group of plants. This is one single genus and you have 5 species. 3 species have a flower structure which is vertical, 2 where the petals are fused and it's horizontal or whatever. So that's our trait data within the species, and this is a complete taxon sample. My aim is to think about what would have been the ancestor state for their flower structure. Vertical, horizontal, fused, unfused, whatever trait data you have, right? How do I go about it? There are two ways to think about it. You start from the tip, go all the way to the root of the tree topology, and then start from the root and come to the tip, and then optimize it. It's called down and up pass optimization. If I start from the tip, I have species B and E. I'm interested in what is the state of F. If I think both traits can have black circle and open circle have equal probability to occur, the optimal thing is 50-50. So half of black, half of open and black circle. Then I'm interested in G. These are my ancestral states. Now F and C will dictate what's the ancestral state for G. You have two black circles, one open circle. What will be the ancestral state? Black circle. Now this G and B will dictate H. Right? B is open circle, G is solid circle. So you have 50-50. Again, G and A will dictate what's I. Right? So when you do this, it's down pass optimization. Now I start from the root all the way to the tip. So A and I will dictate H, which is open circle. Now this H and B. This would be G. G and C will be the F. Right? Is everyone understood this part? Yeah. So what we did, we went from tips to the root, and now we went from the root to the tips. Down pass optimization up, and then take a final consensus. So now I have I, which is open circle, H, I have two open circles, one black circle. So 0.5, 0.5, 1. It's 1. Then I take consensus between these two. I take consensus between these two. So in my final analysis, what I have is I is open circle, H is open circle, G is open and black, F is open and black. Sure. I has only one state. Through the down pass optimization, H had half open, half black, and one open. What would be the consensus between these two? Then I have at G one black circle, one open circle. Consensus. F has half open, half black, half open, half black. So what would be the part? Yeah? So it's a very simple way of thinking about it, but what happens is you do tip to root and then root to T. And this is how you optimize your trait. It could be any trait as we discussed, but this is for the discrete trait. There are other ways within the parsimony which one could use to optimize the trait matrix. But it does not consider branch length. Is it the long branch which has here to this particular species or tiny branch? So where is the ancestor? An ancestor to the tip, how long for a time was there for that particular trait to evolve? That information has not been incorporated in our calculations, right? We assumed it's equal, like we ignored the branch lengths completely. We were just interested in the ancestral state, what was the ancestral state and the daughter species state. But we didn't think about the time. And time may have a role to play, right? If it has a longer time for that trait to change or remain similar, or is it a shorter time? That information we have not incorporated. So likelihood approach does allow to incorporate branching time and a length. So branch length, I'm sure all of you remember from the likelihood tree and a Bayesian tree, you will have branch length, which is the information number of evolutionary changes which that particular branch has. So likelihood approach will use now these branch lengths also when you do the character reconstruction. Also Bayesian approach uses character branch lengths. You can use likelihood approach again for a discrete traits as well as for continuous traits. Important thing is you can also have a model of trait evolution. Do you guys remember model of sequence evolution? You had multiple models. Similarly, for a traits also, there could be multiple models. We just thought about continuous trait could be following normal distribution, it follows exponential distribution or Poisson distribution. Based on that, you will select appropriate model of trait evolution for when you do the likelihood approach for a ancestral trait reconstruction. For a discrete trait, there is only very few models which you can use. One is called MK model, Markov model. But for continuous trait data set, there are multiple models. The simple or a null model is a Brownian motion model, which mimics normal distribution. And an MK model is largely for the discrete data. Yeah. So we'll talk about only discrete data today. So which uses the continuous Markov models for the trait evolution. You must have heard this term Markov models, right? What? Yes, but what was the main assumption when we say it's a Markov process? It assumes the random process. So one of the main assumptions is if I'm reconstructing the trait, it does not have a memory of what is its ancestor has or the tips has. That particular reconstruction for those particular nodes is the information used to think about ancestor node. The rest of the tree could have multiple traits and information, but which is not used when you reconstruct for only that node at that time point. What was the trait? All the other information is ignored. So if you have a binary trait which is 0, 1, what could be the path a trait can take? Either it goes from 0 to 1 or 1 to 0. Right? So you could only have a state which is 0 or 1. And aim here is you have a trait data, which is your observed data. You have a phylogenetic tree which tells you how species are related to each other. Aim is what's the max likelihood your ancestor has this particular trait which is going to explain your observed data. And this is the model which you will use to reconstruct the trait evolution. So you have a tree topology. You can use this. This is a branch length also you want to use. And you are interested in these nodes, right? But in the parsimony, we didn't use this branch length. This can have 0, 0, 1, 0. This, imagine now, either it could be this tree, which is a longer branch, longer time for it to evolve. This is a very short branch versus this is a long branch. And that could influence what is your ancestral trait. Can we incorporate that branch length in our calculation? Is the likelihood method's aim. And Markov models, when they are reconstructing at this trait or this node, it's not influenced what's this node has it. Remember in the parsimony you always went from here to the node. So they don't think here and here, then here and here. So there is no memory of what is the tip when you are thinking about only these two states. Which means that transitions which happens from 0 to 1 are independent of each other. Another assumption of this is the rate of changes across this tree is equal. It doesn't really change in this particular model, MK model what we are discussing, but one can think about it changes and there are other appropriate models for that. And the rates are estimated only from this tree and the data which you are given. It has no other information, right? What other prior information we could potentially provide? Right now we are not and we will not, but what are the possibilities to have that information? For example, if I have a fossil which is which I know where to place in that phylogeny and also trait information. I could potentially use that, but none of the MK models allow you to do that. So this is your tree. You have a trait data, which is at the tip. You are interested in reconstructing what's the ancestral state here, and given these are the branch lengths. How can we use this branch length T1, T2, T3, which may have an influence on what is the character state at this particular moment? As we discussed, we have only binary data, which is 0, 1. So if I have to create a transition matrix, it would be 0, 1, 1, 0 and 0, 1, 1, 1, 0, 0, right? So what is this Q01 says? The rate at which character would change from 0 to 1 and 1 to 0. Do you remember model of sequence evolution? There also you came up with this term called transition matrix, right? Similarly, you will have it here. And then you have these branchlets and you are interested here to reconstruct the ancestral states, right? And aim is to have a maximum likelihood which would explain this observed data with reconstruction of ancestral state with like maximum likelihood score to explain the current data. How do you think about this instantaneous matrix or a transition matrix? You have a 0, 1 trait, D1, D1, D0, 1, 0, right? Because we have only two states. Either species can have 0 or 1. If it has 3 or 4 or more, it becomes complex. But right now you have only 2. Does that make sense to all of you? So what are the different models which we can think of? This 0, 1 is equal, which is all transitions occur at equal rates. You could have all transitions occur at different rate. But for a binary trait data, it really doesn't matter. If you have more trait, like more states, then all these things would matter. Or there is some kind of a symmetric, like 0, 1 occurs more, same rate as 1 to 0, but with a different rate from 0 to 2 or something like that. There is a symmetry or asymmetry in your transitions, which also you can incorporate. And then, like model of sequence evolution, you can compare which is the better fit for your data. Which model is better, which will explain your trait data the best, where ancestral reconstructions are particular. Like either I say that open circle is the ancestral trait which has the maximum likelihood score to explain my observed data. Is that clear? So it incorporates the branch length and it works and operates exactly at same way as how model of sequence evolution operates, right? Because you are also interested here in reconstructing what's the state here and here by incorporating branch length. It's faster and also useful for faster evolving traits. And you can have an estimate of error or variance, especially for the continuous trait data. In parsimony, you can't have that data. But in the likelihood approach, you can have the if I have a continuous trait data for a body length and there is associated error, I can incorporate that also. Like if I have measured 50 individuals for a species, I have an estimate for a body length, but which is associated with the error, I can give that also. And also when I calculate what is the ancestral trait, it will give me probability value, right? And there also I can think about what is the associated error. So the packages or programs you can use, one of the most commonly used is R language, and AC, ancestral character estimation in A, is one of the simplest way to think about it. You have in the Jaja package command is called fit discrete. In Mesquite also you can use the MK model what we just discussed for the discrete trait, And for a continuous trait, I think we have all Brownian motion, OU model and other models also. But most likely people prefer now R because you can visualize it much better. So I can stop here and I'll give you one example in which we have used the trait evolution model, methods. But if you have any questions on this part, we can clarify those now and then I'll move on to the example.
Just like phylogeny, brain parsing method also here also it doesn't take any models, right? But slightly takes the model.
Yes, model of evolution. So right now for a discrete trait, we have NK model where the trait reconstruction is only for that particular trait. There is no memory of what other states have. So NK model works, and for continuous you can have a Brownian motion which is equivalent of random, which models the random process, and there are different variations of other processes which are also modeled. But you have to think about which is the better model for your trait data.
So we approach one trait at a time.
Yes.
We don't use combination.
So yes, you approach one trait at a time. You can also think about if the traits are influencing each other, the association between the traits also, and there are methods to do that. Like if for trait evolution, like whatever trait you see present in the species, is it influenced by another trait? If there are association between two traits and their function. So you have 3 or 4 traits, they could be autocorrelated, and all of them could have a phylogeny which influences the kind of pattern you see.
So that is also possible in what packages?
Yes, yes. In fit discrete, you can have that data.
And if it is continuous, there is an option to incorporate standard error.
Yes, yes. And for a continuous trait data also, R and Mesquite both can handle, and a same set of packages. So one of the command will be, is it what sort of data type you have? You have to say that discrete or continuous and the data frame associated with it. For Mesquite, it will be Excel sheet. Huh?
I just would like to know if there is any paper or something like that where we can refer. They have
Yes,
used
yes.
the auto-correlated traits.
Traits, I can circulate something. Yeah, so I'll discuss our example in which we have used ancestral trait reconstruction. This is the work led by Nikhil, who was a PhD student at NCBS, where we were interested in understanding how color defense strategies have evolved in butterflies, but not in the adult butterfly, but in their ontogenic development, where their selection pressure could be different and their responses at each development stage could be different. What do we mean by that? So this is the group of butterflies we did the character evolution. These are called papilionids. These are big swallowtail butterflies. They have they are species in the tropics, but they have a global distribution. They occur in temperate and tropical areas. One of the most species group, very colorful butterflies. They participate in mimicry. Have you guys heard about Batesian or Mullerian mimicry? So Batesian mimicry is where a species mimics another species which is poisonous, but don't produce those toxins, but just mimics the color patterns, and which are warning colors, but without producing the toxic compounds. In Mullerian mimicry, species converge to a color pattern which is warning coloration, but both produce toxins as well as color patterns. And in papilionids, you have both. You have Mullerian and Batesian mimicry. So people have been studying these color patterns and mimicry in adult butterflies and trying to think about what's the role of natural selection through predation. What we wanted to think about, these are the different life stages butterfly goes. This is egg, early instar larvae, late instar larvae, pupal stage and adult. It's not like the adult shows, for example, Batesian mimicry, all of them would be showing the same defense strategy. They use a very different set of defense strategies at each life stage. So what are the traits, life history traits, or what they feed on, or where they feed on? Do they play an important role? What color defense strategies these pupae or early star or late star larvae will use? So that was the aim. Every life stage is under different kind of selection pressure, and they respond to that, and then that dictates what color defense strategy that particular species at that life stage will show. What do we mean by that? These caterpillars could be aposematic, where you have a warning coloration associated with toxic compounds. Caterpillars could be masquerading, which is like they look like a bird dropping, or they show crypsis or cryptic coloration, where they merge with the background. Pupae also show these color defense strategies. So clearly at each life history stage, which is caterpillar and pupae, they could participate or show these different defense strategies. What is the other important factor which could also influence? These two stages, caterpillars are mobile, but pupae are immobile. Would that also affect what color defense strategy they use? Like if I have to escape from predation pressure, where I'm mobile, versus if it's an immobile stage, would that differ? What do they feed on? Is it a toxic plant where the caterpillar feeds versus non-toxic plant? Because butterflies don't produce those toxins, they sequester those toxins from the host plant. So this would dictate if you have aposematic color pattern or not, because they have to feed on toxic plants. Growth type, do they feed on a dense vegetation versus sparse vegetation? Predation pressure would differ, right? Predators seeing the caterpillar would be different on the background on which these caterpillars or pupae are sitting or feeding. So that could also influence what color defense strategy at that particular stage species uses. Also, which part of the globe you are in. Biogeographic history. Are you a tropical butterfly or a temperate butterfly? That could also potentially influence what color defense strategies you employ. So we wanted to see how these things have evolved in these Papilionidae butterflies. So through literature, Nikhil collected all the data, like for each species, what was the color defense strategy? Were they showing masquerading or aposematic coloration or crypsis? Early stage, late stage, pupa, what was the host plant on which species were feeding? What is the growth type? Was it sparsely vegetated or a dense vegetation, as well as was it in the tropic or a temperate? We collected this data at a genus scale. So within Papilionidae, what we had was 23 genera, and we took representative of each of that genus, constructed molecular phylogeny based on multiple mitochondrial and nuclear markers, but our trait data was consistent within the genus. So our unit of analysis here was a genus. Because if within a genus all species would show aposematic caterpillars, right? So that becomes your unit of analysis. In the genera where there wasn't, we used all the available data, because that would influence how we reconstruct our traits. If there is a variation within the genus, then I have to use the species level information. Here, within the genus, trait data was consistent, so our unit of analysis was genus. So after reconstructing this tree, we had collated this trait data. Our aim was to reconstruct the ancestral traits. So we used the Bayesian method or SIMMA, where we haven't discussed that, but we also use the likelihood method with equal rates. So the stochastic mapping, the advantage here is, remember we said we are not incorporating phylogenetic uncertainty. We have one tree topology, which is the input tree, and branch length, which is depicted in that tree topology, will be taken. But if there is a phylogenetic uncertainty, how these genera are related to one another itself, and I want to incorporate that, then I can go for a stochastic trait mapping, where multiple tree topologies will be used to reconstruct the trait, ancestral traits, and then you will have a consensus tree. But for a likelihood and parsimony, you give one single tree topology. So this is the result for early instar, which are the early caterpillars stage, where they are little smaller. What you see, orange depicts aposematism, green depicts cryptic, and masquerading is in blue. It's not like aposematic has evolved once, it has evolved multiple times. you see some sphere genera show masquerading, some shows crypsis, and some show aposematism. Not a very clear picture which was the trait which is ancestral trait. Aposematism has highest probability, must be around 0.7 or something. But you can also see some blue and green here at ancestral state. Then we mapped late instar. Again, 3 strategies which they could potentially use: aposematism, cryptic, and masquerading. Interestingly, all the ones which were showing masquerading here now showing cryptic coloration. So when they become bigger, their defense strategy changed. When they were smaller, they could afford the variation in their defense strategy. Now all the bigger ones were showing cryptic coloration. What happened when we looked at pupil? They're all cryptic. There is no masquerading and aposematic coloration. By the way, these same caterpillars were showing aposematism, so they have fed on toxic plants. But at a pupal stage, they went for crypsis. Because if you remember, early instar and a late instar, at the caterpillar stage, they move, they are mobile. Pupae are stationary. So cryptic strategy had an advantage. And it's not like in individual species you see cryptic has evolved. The ancestral state is cryptic coloration. So all of them show this particular trait because their ancestor had it. And no selection pressure which had changed this particular trait throughout their evolutionary history. Right? This is where it's useful to look at in a comparative data. People have looked at where did we got this idea. People have studied individual genera and species and selection pressure at each stage and shown that, oh, at a pupal stage they are cryptic, at early or late stage they are masquerading or aposematic. But now when you look at all of them together, whatever, 60 million year evolutionary history, you see cryptic coloration is the ancestral trait. All of them have it.
We cannot actually say that there is no selection pressure for this
There is.
other stage. That itself, I mean, the
Yes,
trait
that
itself
also,
is
yes.
correct.
Then we wanted to look at what's the role of, is it the interaction of what plant do they feed on and also the visual background. Is it a sparsely vegetated tree or bush are they feeding on, or a dense vegetation? And before that, is it a toxic plant or non-toxic plant? So toxic plants are marked in yellow, non-toxic plants are in black. If you just visualize this data, you could see non-toxic plants was the ancestral trait, with 0.2 or 0.18 was the contribution of toxic plants. So the ancestral trait here was non-toxic plant. Multiple times feeding on toxic plants was evolved. But then we were also interested in which ones were feeding on sparse vegetation and dense vegetation. So wherever you have aposematic coloration, invariably it's sparse. Wherever you have dense vegetation, it's cryptic. So clearly, on what background, visual background for a predator has a role here. So now we don't know now this. All toxic plants and their habit itself is sparsely vegetated. So that's why this caterpillar can sustain, and all dense vegetation is non-toxic. That is something very different question. But here what you see is species which feed on dense vegetation are always cryptic, except few species here. Rest of them, whenever there is aposematism, they feed on toxic plant and they are always sparsely vegetated. Is that clear? Like now we are looking at multiple traits, how they may influence each other, and if there is any interaction. So species which are aposematic at a caterpillar stage, they are feeding on toxic plant, and these toxic plants are always sparsely vegetated. So the visual background, apart from the phytochemistry of that plant, is also equally important. Is everyone okay with this?
I have one doubt, like maybe I'm not that clear with that, but different stages they show different
Different
or
strategies.
right?
Yes.
So in the different stages, do they have different predators?
Not really. So the predators remain, which are largely birds. So even for adults is largely birds. So one of the main, I will not say only birds, but one of the main predator group is birds throughout their life cycle.
But like, maybe I'm wrong. One strategy throughout their life cycle, do you think?
But they are not showing that one life. So that's something you will think, if the predator doesn't change necessarily, will they have a single strategy? But their life history traits at each life stage are very different. So adult is a size of 5 millimeter. For example, caterpillars are of a different sizes, shape, right? Adults can fly, larvae can walk and move, caterpillars can move, pupae are stationary. So each life history stage has its own constraint. They are mobile or immobile, right? Size matters, where they are located also matters. So visual background, where they are sitting and feeding also matters. And then all of them are dictating what defense strategy that particular species is showing.
It means they have more chance of getting predated by during the adult stage, right?
So the predation pressure, does that change over life cycle is a different question, which we haven't addressed here at all. How the predation pressure changes. But if one assumes the predator guild is similar, what are the defense strategy? And is it because of the predation pressure itself is different at each stage? That also affects what strategy you use. It's a different question. But that we really can't talk about it. But there is a predation pressure at different stage, and these are the life history traits which are used to dictate.
So, can you run multiple traits at a time?
So when we did this study, no, we didn't. We individually went one by one trait, but now you could. You can even have a model in which you have these traits and their interactions and then run that.
So for this, what you did, like add on this trait of the other traits on this line.
Yes, and you use the association. So we use GLMs to look at the associations of those traits.
So that was done separate.
Separate.
But now you can.
Yes, you can do it.
I have a question. Suppose in a genus, most of the species, they have one particular state for the character, and only few have the other state. So how do you code that character for the?
So you don't. Then you need to look at a species level analysis. So when it's consistent within the genus, then only you can use genus as a unit of analysis. So if, for example, if I'm looking at butterflies called bird wings, have you seen? Troides. Okay, so I'm looking at bird wings as a group of butterflies. It's one genus. Within that genus, this character is consistent. All caterpillars show this. Then genus as a unit of analysis I can use. But within that genus, if some species show very different characteristics at a early stage and a pupal stage, then my analysis has to happen at that stage. And what stage am I interested in looking at the ancestral trait reconstruction? So again, in that you can think about it. So if there are 50 species, only 2 species show some different and 48 have one character. Then you can give a proportion and think about it. But if you have very variable trait within that group, then you will do the analysis at a species level. You won't do it at a genus level. So that would dictate what trait are you looking at, what's the appropriate scale to look at.
So here the adults, they show sexual dimorphism.
Not all. Adults show very different defense strategies. Not all species show sexual dimorphism. Some show Mullerian, participate in Mullerian mimicry, some are in Batesian mimicry. We haven't done that analysis. If you have a different color defense strategies as pupal and a larval stage, what's your adult strategy? And that would be interesting to do, but we haven't done that analysis.
So similar to that, I think, can we check for the epigenetic things also? So how does that influence is it temporary, these changes in the temporary?
Yeah.
Metabolically speaking, switching from one, say, coloration to another or developing one character to another would require a lot of metabolic turnovers. So
Yes,
could be
people
I mean,
looked at
could be epigenetic.
no, but it's in a species. If you see it consistently, there is one kind of a pathway which species is following. What's the mechanism of that? It's a different question altogether.
That's a different field.
Yeah. So the idea here is you can use ancestral trait reconstruction to understand a question where multiple species of these butterflies, when they participate or use these color defense strategies at different life stages, is it at each lineage it's a unique or because of their shared ancestry, right? This is looking at the entire clade of Papilionidae butterflies. So this is one family of butterflies. Where all pupae showed cryptic, early and late stage showed a very different color defense strategies. Within that smaller, you had multiple, all three were used. When you become little larger caterpillar, you had only cryptic and aposematic, masquerading wasn't there. So the color defense strategy is also reduced, and it was also influenced by what plant you feed on and what's the habit of that particular plant. We had no effect of biogeography, like is it a tropical or a temperate species. Did they show very different set of color defense strategies? We didn't find any evidence for that. Yeah, so we tried to synthesize this as a feedback loop. So basically you have these different stages, small and adult caterpillars, pupae, but at each life history stage, you have a different selection pressure and which reflects in different color defense strategies. How did we use? We used ancestral reconstruction to show what was the ancestral trait for this group. At a family level, we had the data at a genus level, right? And then the association between these traits. And these are the different workshops and their material which I have used in my earlier talk, and all the links to that. So you guys can go back and check some of the material, especially for a correlated traits. How do you look at it? So all the links are here. You can go back and look at it. That's it. Any questions?
So, like you said that if there is a genus which has variable traits, then you go to the next like little species level you do the analysis. Now, let's say all those genus that you have used. So I look at 50 genus, and out of those 50 genus, 48 show that there is conserved traits, but 2 of those genus show that they have multiple. So what do we do in that?
So you can think about multi-state characters for those 2 genera. And then think about it, how that has been. So the 48 are much more consistent, these 2 are different. So you can think about a ways in which so it depends on what trait are you looking at, what's your question, and what is the most appropriate scale at which you want to answer that question.
Yeah, I also had a question. In your ancestral trait reconstructions, it's all dependent on the state at the tips, right? So
branch lengths.
And the branch lengths, yeah. So is there a way, because a lot of these phylogenies don't really sample all the taxa, right? So the sampling also can have an effect on the...
So assumption here is that you have sampled possible, like if I'm looking at Papilio as a genus, I know what's within the genus variation is, unless and I'm confident that all of these species show this particular trait, I wouldn't go.
No, but is there a way we can account for...
Yes, there are ways. Yeah, you can. So now there are ways in where you can give a proportion of sampled lineages.
Correct. Yeah,
but we didn't do that.
Okay,
yeah. But there is a way to do it. Are there any questions? And in the practical session, we'll show you not R, but in Mesquite, how to enter the data and how to do reconstruction using parsimony method.
Okay, thank you, Jahnavi. So we'll, in the practical session today, she'll be there, so we can talk to her.